Hi, it's Dr. Josh with Alzheimer's Breakthrough, and I just finished interviewing Dr. Mark Mattson. What an amazing researcher, scientist. I love what he said at the end, which is work hard and have fun. We definitely had fun at the interview. You're going to want to watch the whole episode. We definitely dive deep into some of the biochemistry, but we also have some important tips along the way of specific lifestyle approaches you can take to prevent and reverse Alzheimer's and other neurologic brain disorders. Dr. Josh, Peace and Plants, out. Dr. Mark Mattson is one of the world's leading neuroscientists in the fields of brain aging and neurodegenerative diseases. He is a former chief of the Laboratory of Neurosciences at the National Institute of Aging and a professor of neuroscience at Johns Hopkins University. Over the past 3 decades, Dr. Mattson has made landmark contributions to our understanding of how dietary and lifestyle interventions, particularly intermittent fasting and exercise, can protect the brain, enhance synaptic plasticity, and delay the onset of diseases like Alzheimer's and Parkinson's. He has authored more than 500 peer-reviewed articles and has been named one of the most highly cited researchers in the biomedical sciences. Dr. Mark Mattson. All right. So it's Dr. Josh with Alzheimer's Breakthrough. And what if skipping a meal could help save your brain? Today's guest, Dr. Mark Mattson, is not just a top-tier neuroscientist. He's also a pioneer who has uncovered how intermittent fasting and lifestyle changes can protect your brain from Alzheimer's. We dive into groundbreaking science, debunk myths about aging, and explore simple, actionable strategies you can start using today to boost cognition and extend your health span. Dr. Mark, thank you so much for joining us today.
Welcome, Dr. Josh. It's nice to meet you and talk with you.
Likewise. And what I followed your work for over 10 years, so it's really exciting to meet you on this forum. And then also, this is an Alzheimer's broadcast, and you've been one of the pioneers in doing research on Alzheimer's. So why don't we start there, if that's okay?
Yeah, well, I could give you a little bit of this kind of a quick timeline on my career, because I started out as studying the effects of stress on the neuroendocrine system that controls stress, the brain, adrenal gland, cortisol system. And then my PhD work was on a stress-responsive neuroendocrine system that controls molting in crabs. Well, it's kind of interesting because it's kind of analogous to the hypothalamic pituitary adrenal system, stress system, and that there's these neurons in the brain of the crabs that they tonically inhibit the production of the molting hormone. And then when the crab gets ready to molt, it goes into essentially a hole. It hides and removes itself from the stress of potential predators. And when that kind of chronic stress is relieved, then it molts. And then my postdoc work, which was in Colorado, I studied developmental neuroscience, studying how neurons grow their axons and dendrites and form synapses, sites of communication between the neurons. And I discovered that a neurotransmitter, glutamate, which turns out to be the most important neurotransmitter in our brain that a lot of lay people haven't heard of, but 90% of the neurons use glutamate, an excitatory transmitter, as their neurotransmitter. So 90% of the neurons in the brain, the main circuitry uses glutamate. And I discovered glutamate is critical for the wiring of the neural circuitry during brain development. Then I got interested in brain aging because it turns out if you stimulate neurons too much with glutamate for too long a time, it can cause damage to them and even kill them in a process we call excitotoxicity. And it turns out, so that got me interested in death of neurons, and which occurs in Alzheimer's. And then, so I pursued that actually through my whole career, provide a lot of evidence that with aging, normal aging, and much more so dramatically in Alzheimer's disease, there's an imbalance in the regulation of neural network activity and excitatory imbalance. So we think that in Alzheimer's disease, the damage to neurons involves this excitotoxic damage. And this will become relevant to our discussions about intermittent fasting and exercise and how they may counteract Alzheimer's.
Very cool. Well, the suspense is rising. Why don't we go there right now? Talking about, well, first of all, for the audience, glutamate. People may have heard of glutamate. It's an amino acid. People may have heard it as like MSG, monosodium glutamate, is sometimes added to food. So it's around us, definitely.
Yeah, you're right. It's the building block of proteins, which is interesting from an evolutionary perspective. You have this amino acid that it's involved in the structure of cells and all their functions, but then it's also packaged in the neurons and released discretely at synapses between the neurons. So in the brain and elsewhere, outside of the cells, glutamate levels are very low because there's this active system that removes glutamate and gets it into the cell. So the reason monosodium glutamate doesn't kill our neurons is because of this efficient way our cells take it up and prevent it from accumulating outside the cell. I actually wrote a book. So since I retired from the National Institutes of Health, National Institute on Aging, about 5 years ago now, I wrote 2 books. One's the Intermittent Fasting Revolution, which is on the science of intermittent fasting. And then the other, and this focuses quite a bit on the brain, actually. And then this one, Sculptor and Destroyer. So the title of that book, it's about glutamate. So the title is, during brain development and in healthy adult brain, glutamate sculpts the neural circuits. But then when things go awry, whether it's a chronic neurodegenerative disorder or something like a stroke or traumatic brain injury or epileptic seizure, which is excess activation of glutamate receptors, that's the destroyer part. So,
right. And of course, it's very complicated, and it's kudos to you for working out a lot of the basic biochemistry and neurobiology behind it. And you teased us a little bit about that there's a role of glutamate both in fasting and also in exercise. What is that role?
So the first paper we published on intermittent fasting, and this was when I was at University of Kentucky. I was a professor there for 11 years before I moved to the NIA. Right, so we're interested in brain aging, and it was even well known at that time in the 1990s that calorie restriction or energy restriction in rats or mice extends their lifespan a lot, particularly when it's begun when they're young and then throughout their life. And there are 2 ways that this was done. One was to, every day you give the animal less food than they would eat spontaneously, right? And you can control it like 30% calorie restriction. So the other way is every other day food deprivation or every other day fasting. So 24 hours, no food. The next 24 hours, they can eat what they want. In both cases, the animals live up to 50% longer, depending on the strain of the rat or mouse. And so we just kind of decided on every other day fasting or food deprivation. And we had, at that time, we did not yet have our genetic models of Alzheimer's that have amyloid plaque accumulation in their brain and the intraneuronal so-called neurofibrillary tangles, tau tangles. But what we did have was an excitotoxin model where there's a lot of interesting stories in all of this, which I've I've told in this book on glutamate. So there was an incident in Canada this a long time ago. I think it was the 70s or something, where several people that had eaten at the same restaurant within the next day or two, they developed amnesia. Okay. And it was like very profound.
Sure.
And so it was traced back to some shellfish they ate. They all ate the shellfish. And it turns out it was kind of a drought year where they were getting the shellfish from. And the shellfish feed on algae. And the algae had accumulated this excitotoxin called canic acid or canate, which turns out to be a very potent activator of glutamate receptors. And more importantly, cells are not able to remove it and take it in, get it away from the receptors on the outside of the cell. So it just keeps stimulating the neurons like crazy. I should mention these people had seizures too.
Wow.
Epileptic seizures. So what happened was that this excitotoxin damaged neurons in their hippocampus, a brain region that's very important for learning and memory. It actually wiped them out. One of them died from it, and they actually looked at autopsy. So we and others had found if you administer this canic acid to rats or mice, it can cause seizures, epileptic seizures. It can damage the hippocampal neurons. It can consequently impair their learning and memory, which we can test in maze. So we found if we maintain rats on intermittent fasting, and we had to maintain them on intermittent fasting for several weeks, actually a month before we saw clear effect. But by then, we found that the neurons in their hippocampus were protected. They didn't have the seizures, and their cognitive function was retained compared to rats that were fed ad libitum, that were not on intermittent fasting. So that was the first demonstration of a neuroprotective effect of intermittent fasting. And then we went on to we and others developed mouse models of Alzheimer's that have amyloid and tau and cognitive impairment, memory problems. And we showed that long-term intermittent fasting over a period of, actually we did a year, because in these mice, it takes, at least the mice we developed, the pathology and the cognitive impairment develops in an age-related manner. They get progressive accumulation of the amyloid, progressive neurofibrillary tangles, and cognitive. Yeah, so that's kind of in a nutshell, involving scores of graduate students and postdocs and technicians in the lab over a period of about 10 years.
Wow. So that's intermittent fasting. What is the roles of glutamate with exercise?
Yeah, well, I should mention something about the mechanism. So then the question becomes, what is it that intermittent fasting is doing to protect the brain cells? And kind of the two main things we found were it enhances the action of an inhibitory neurotransmitter called GABA. So it protects the neurons against excitotoxicity, hyperexcitability, by quieting them by activating inhibitory receptors. So GABA receptors are a target for the first drugs that were used to treat anxiety disorders. Valium, diazepam. And then the other thing related to that, it's been known for a long time, before we even started working on fasting, that ketogenic diets can be beneficial for people with epilepsy that don't respond to drugs. And both the ketogenic diet and ketogenesis from fasting, which in my view is healthier than eating a lot of high-fat food and not much plant-based food. Both ketogenic diet and intermittent fasting enhance the GABA inhibitory tone. That's why they suppress seizures, and that's, at least in part, we think why they're protective. The second reason we think that intermittent fasting is beneficial for the brain is that it stimulates the production of a protein called BDNF, brain-derived neurotrophic factor. And BDNF is known to protect neurons against a range of stressors, whether it's excitotoxic stress or metabolic stress, like mitochondrial impairment, for example. But BDNF is also important for learning and memory. In fact, in mice where you can genetically reduce BDNF levels, they have problems with learning and memory. Interestingly, they also develop obesity and diabetes because BDNF acts in the hypothalamus, kind of like these GLP-1 peptides to suppress appetite. So that's kind of the interesting story there. So exercise, and there's a lot more there. I mean,
Sure.
We're going
Right.
slow.
Right. We could talk for hours about
Yeah.
one small part about BDNF, for example. But
Yeah.
just from a general overview,
Okay.
what's the exercise piece, and then we can delve in. Sure.
And so the thing I haven't mentioned too is epidemiological studies. So there's really good evidence that people who are chronically overweight, particularly with insulin resistance, and certainly with frank diabetes, if they're in that poor metabolic state, I call it metabolic morbidity because it's, in fact, obesity is now considered a disease. So chronic metabolic morbidity is a risk factor for definitely stroke, but also Alzheimer's disease and vascular dementia, which is another type of dementia. So that's based on a lot of epidemiological evidence. So you can go from, the human epidemiological evidence, then you take in the animal evidence. If you make mice or rats obese by giving them high fructose corn syrup and, load them with saturated fat, that's not good for their brain. So it kind of does the opposite things I've talked about with intermittent fasting. Then exercise, it's similar. There's good evidence that people who exercise regularly, their brain functions better longer as they age. And I think anyone who exercises regularly realizes this, particularly if they have an injury or for some other reason can't exercise. Dramatic effect on mood, right? But also cognition. People generally, their brain is functioning at a higher level when they're physically fit. And the other thing with fasting is that, and this is something that's it should be looked at a lot more. My own personal experience is that, and this started kind of by serendipity. When I was a graduate student at the University of Iowa in the early 1980s, I had some problems with gastric reflux. Okay. And at that time I was eating breakfast, and I'd often have whatever, orange juice, toast, egg, whatever, cereal. And then I lived 7 miles from the lab, and so I'd ride my bike in and back. So I get up in the morning, get on the bike, and ride in, and then my gastric reflux was a lot worse because I have this. I mean, it seems kind of obvious, but I have this food in my stomach, including orange juice or whatever, and then I'm bending over, and I'm riding 7 miles. So anyway, I just found that if I didn't eat breakfast, it had a beneficial effect on my reflux. And so I haven't eaten breakfast since then.
Oh, wow.
Okay. Because I also found, and then it became I found that my brain was working better in the morning. I was thinking more clearly in the fasted state after I hadn't eaten anything since 6 or 7 the previous night. And then, so I just kept that routine. I get up early, get all my mentally challenging things done in the morning while my ketones are up. And then when you eat lunch, as you know, you feel a little sleepy.
Sure.
Which is kind of the opposite of your brain performing at its highest level. And Yeah, and then I also exercise in the late morning before I eat, and I compress my eating window to about 6 hours every day. So in general, people have a metabolic switch from glucose to ketones, from glucose in the liver to ketones coming from fats. After about 10, 12 hours of fasting, the ketones start to go up. And then the longer you fast after that, the more they go up. And then by a couple days, they kind of stay up like 10 times higher than they would in basal condition.
Wow. And so you just mentioned kind of switching from glucose burning to ketone burning. You have a recent paper in Nature Metabolism talking about this whole concept of cyclic metabolic switching. Can you unpack that for our listeners and how that
Yeah,
might actually be relevant for Alzheimer's or brain health, et cetera?
yeah. So with exercise and intermittent fasting, both, particularly people who do like endurance exercise, and I think this is just a hypothesis, but there's educated guess that what endurance athletes may be previously referred to as second wind, right? You've heard that phrase before. Like you start out, and I noticed this too, actually, when I do a lot of trail running, for an hour, hour and a half. And I noticed that like my performance would start to improve after about half hour or 40 minutes into the run. Okay, so that's the idea of second wind. I think what's happening is if you start running or whatever, biking, and you still have glucose in your liver, which is used before the fats are mobilized from your fat cells, that glucose is depleted as you're running or biking. And then when it gets depleted, it's switched to ketones. And then once the ketones start going up, this is, again, hypothesis, the performance goes up. So this cyclic metabolic switching, exercise involves cyclic metabolic switching. You have a metabolic stress or challenge. It's a stress on your muscles, your cardiovascular system. Even your brain cells are more active, and their metabolism increases some during exercise, which kind of makes sense from an evolutionary perspective. If you're out in the wild, say hunting a prey animal or something, your brain's got to be working well as your body is expending effort. But anyway, the point is, during the challenge, the exercise, it's a stress, but a good stress on your cells, as long as it's not too intense for too long. And then the same with fasting. During the fasting, it's a mild stress on your cells. Okay, and those cells, during the challenge of exercise or fasting, cells adopt a stress resistance, conserve resources mode. So they upregulate pathways that bolster the cell's resistance to more severe, potentially damaging stress, like free radical damage, oxidative stress, which increases a lot with exercise, by the way. Metabolic stress, the mitochondria are working overtime, and then the other thing is a process called autophagy is stimulated. It's the cell's garbage disposal. Our cells are the proteins, the membranes, the fats or lipids, and even DNA in our cells is constantly being attacked by free radicals. There's effective repair systems or ways of lessening the free radical damage. And one way is our cells have antioxidant enzymes that can be upregulated by exercise. We found can be upregulated in brain cells by fasting. Autophagy with extended exercise and fasting increases. Okay, so that's the challenge part of this cycling. So we could call it metabolic cycling,
Okay.
between the metabolic challenge and recovery phase. Or another paper I published with a colleague of mine who works on a concept called hormesis, which you may have heard of, but it's just, if you subject this...
And I see a book on hormesis behind you. It's on the right of my screen, but
Yeah, that was a long time ago. It's an edited volume. The books I wrote are like me.
Okay.
But this is an edited volume with chapters by different authors. But me and Ed Calabrese were the co-editors on it. He's done a lot of work on the concept of hormesis, which you get kind of a biphasic response to a potentially toxic chemical or potentially damaging situation like exercise or fasting. If you fast too long, you starve to death. So in that paper, we called it catabolic anabolic cycling. So far I've talked about the challenge phase or the mild good stress phase. Then you need a recovery period. So this is well known with exercise. Your muscle cells don't get bigger while you're exercising. Their endurance doesn't increase while you're exercising. Muscle cells get bigger, stronger, more mitochondria during the resting, eating, sleeping phase. We think the same is true with fasting. During the recovery period, cells go into a growth and plasticity mode. So you have the switching between stress resistance and growth plasticity mode. But if you don't have the first, the stress, the challenge, the things aren't set in motion that enable the good things to happen during the recovery period. Yeah.
That makes sense. So going back to the switching from glucose to ketone metabolism, and actually before we go there, explain to the audience what ketones are.
There's fats in your fat cells, mainly triglycerides and diglycerides. Okay, and that's your energy storage that can let you function well for days, weeks, months, even depending on how much fat storage you have. You can go a month or more without consuming any calories. And so that, however, if you eat 3 meals a day, particularly if you have carbohydrates, there's another energy source that's carbohydrates. And when you eat carbohydrates, they're broken down to glucose, to sugar. And sugar goes into the liver where it's accumulated in the liver cells in a form called glycogen. Okay, so glycogen is essentially glucose in the liver. And the liver has about 700 calories worth of glucose. And that is used first. So if you eat breakfast, lunch, dinner, and have carbs at each of those meals, and you don't exercise, then you may never deplete the glucose stores in your liver because every time you eat, it replenishes them. Okay, so then with fasting, if you fast for, again, 10 to 12 hours, which is like about 700 calories, approximately, the liver stored glucose, that glucose is used up. Then what happens is these fats in your fat cells are broken down into shorter fat bits called fatty acids. They're released into the blood, and they're taken up into the liver cells. And the liver convert them to 2 ketones, what are called ketones. Acetoacetate is one, and beta-hydroxybutyrate, which is one we and a lot of other people have studied the most. Because so during fasting, the reason people can live for a month with no calorie intake is the ketones are supplying the energy to the cells that would have otherwise been supplied by glucose. Okay? So the ketone's very important energy source, but at least the beta-hydroxybutyrate ketone, BHB, it acts as a hormone. And I talk about this not in a lot of detail, but in this cell, was it cell metabolism or nature metabolism? I don't know.
think it's nature metabolism. I could be wrong.
That's recently.
But isn't this BHB? Don't you call it like a fastokine?
Yeah. I call it a fastokine. It's produced in response. It's produced by liver cells in response to fasting. It's released into the blood, then it's distributed to cells throughout our body and brain, and it acts as a hormone in that it will affect, it will stimulate certain genes, turn on certain genes. One is BDNF. My lab and the lab up at NYU at New York University kind of simultaneously published at the same time the same result, that this BHB will stimulate BDNF production in neurons. You can expose cultured neurons directly to this ketone BHB, and the BDNF gene will be turned on. And then, actually, that was a postdoc in my lab, and she worked out the mechanism. And then a lab up at NYU showed this is true in the brain in living mice too. So we think that BHB may be part of the reason that fasting will increase BDNF levels in the brain. So it's acting like a hormone, so there's this whole classes of things that are called exerkines and fastokines. A lot of work in the exercise field has been on molecules released from muscle cells in response to exercise, which you can call them exerkines, or some people call them myokines. There's some over myo means muscle. Kine is just something released into the blood. It's essentially similar to a hormone in the idea. But with the exerkines, there's been a lot more done actually with exerkines. One of my colleagues at NIA, Henriette von Prague, she discovered, actually I recruited her to the National Institute on Aging because she had discovered in another lab at the Salk Institute that exercise, running wheel exercise in mice or rats will stimulate the production of new nerve cells from stem cells, at least in one brain region called the hippocampus, which I mentioned. That's called neurogenesis, the production of new neural cells. And so then, when she came to the NIA, she followed up on this, explored the mechanisms, and she discovered a new, never previously described exerkine that's released from muscle in response to exercise and stimulates neurogenesis. That's called cathepsin B. And there's a whole literature on this. I mentioned it briefly in that article, but there's more comprehensive articles people can find. And anyone listening to this, don't just Google things. If it's science, biomedical science related, go to PubMed. That will lead you to the actual science publications. And if you're interested in like getting an overview of it, just put in like exerkines review, or if you want to limit it to a top journal, you put in exerkine review. Well, you have to know a little more detail, but Nature reviews are just NATREV, and that'll be a review in one of the Nature's reviews journals. But anyway, I'm a little worried about what's happened with the social media and the internet and the proliferation of influencers who aren't necessarily experts in the field and are trying to dramatize things, sensationalize things, and exaggerate things that haven't been shown or may have a minimal or no effect.
I agree.
And so the reason I agreed to be on your podcast, because I know your MD is up on the literature, and, we can talk about things that are generally well documented.
You need to hydrate every day, right? All day, every day. Why not make a quick, healthy change to your hydration and use that as an opportunity to get more micronutrients into your body? That's the idea behind hydrating with purpose, and that's what our friends at Organifi are all about. They make these superfood powder blends that you mix right into your everyday water, and boom, it transforms into a delicious drink that floods your body with power from nature, like their green juice. I know there are a lot of green juices out there, but this has been my favorite for years now. It tastes great, which is already amazing because most green juices taste okay at best. I think a lot of people would drink this just for the taste alone, but it's better than plain water. And then you've got all these amazing ingredients from all over the world, including chlorella, spirulina, moringa. These are ingredients that pack a ton of nutrition into a tiny little package. That's how just one scoop a day can make a huge difference for your entire body. Plus, each serving has a clinical dose of the adaptogen ashwagandha, which has been shown to help balance cortisol levels. Cortisol, as you probably know, is your stress hormone. It's the fight or flight response that's supposed to help you escape from a bear or a tiger or lift a truck off of a family member. But you also get that excess cortisol when you're just stressed out and there's nowhere for it to go. That means we're just stewing in too much cortisol, and that's really bad for the body. It messes with your weight, it messes with your sleep, it messes with your immune system, it messes with just about everything. And lately, maybe now more than ever, we're all dealing with way too much stress. A glass of Organifi green juice in the morning can be a huge help in times like these. So Organifi sources the highest quality ingredients from organic farms worldwide, ensuring that their products are vegan-friendly, gluten-free, and free of any artificial additives. Plus, it's glyphosate-free. It's pure nutrition in every sip. Now, here's the best part. Since you're a listener of the Alzheimer's Breakthrough with Dr. Josh podcast, you get the VIP treatment. When you visit Organifi/Dr. Josh, that's ORGANIFI.com/DRJOSH. You take a look at their collection, and when you go back to check out, use the promo code Dr. Josh, DRJOSH. You're going to get a 20% discount on all of their products. So go ahead and check it out. Find out how these superfoods can help you on your health journey today with Organifi at Organifi.com/Dr. Josh. Use the promo code Dr. Josh, DRJOSH, at checkout and try it for yourself. Your satisfaction is guaranteed. You talked about, and you wrote a whole book on glutamate as an excitatory neurotransmitter. And then you also mentioned GABA, which is a calming neurotransmitter. One thing you didn't mention, at least here, but is there's actually an enzyme in the body, in the brain, that can convert glutamate to GABA. It's called GAD. And there's things that can block that conversion, like black mold toxin, trichothecene. I'm just curious, have you studied that enzyme GAD, and has that, I mean, it's just fascinating that all these things are interwoven and complicated, but woven together. What are your thoughts?
Yeah. Now this is a whole interesting story here. And it also links energy metabolism to these neurotransmitters, glutamate and GABA, because nerve cells are not the only cells in your brain. There's other cells called glial cells. Astrocytes is the most abundant one of those. And so astrocytes produce a chemical called glutamine, which is involved in energy metabolism, but they can release that, and that glutamine can be taken up by neurons and then used to produce glutamate, okay, in the excitatory neurons. But the inhibitory neurons that use GABA as their neurotransmitter have this additional enzyme, GAD, or glutamic acid decarboxylase. Just kind of some basic science here. If you hear something like glutamic acid is the same as glutamate, the aspartic acid is the same as aspartate, et cetera, et cetera. But anyway, so I didn't know that this toxin produced by mold inhibits GAD. So does it cause excitotoxic damage?
Correct.
Or hyperexcitability?
So when I take care of patients who've been exposed to black mold toxin, the trichothecene, often they'll be anxious, they'll be wired, they'll be tired, they'll have elevated glutamate levels, low GABA levels, and one of the mechanisms is the knockout of the conversion of one to the other. I've seen that clinically in hundreds of patients. I haven't written a paper on it, but yeah, it's just so it's amazing how these simple molecules that you look at basic biochemical pathways, but it's amazing how it all comes together clinically sometimes.
Yeah, and so the ketones are increasing GABA and suppressing anxiety, right? Exercise and fasting are good anxiolytics. We actually found in mice that we can test their anxiety level, and it's reduced when they're adapted to intermittent fasting. So that would be a good prescription for these patients with the black mold, get their ketones up, exercise. Well, it's a good recommendation for almost any ailment, I guess.
Correct. And let's talk a little bit more about ketones, because, traditionally, if you talk about nutrition and macronutrients, you've got protein, you've got fat, you've got carbohydrates or sugars. Do you think ketones should be kind of a fourth macronutrient? What do you think about that?
The ketones aren't generally consumed in the diet, so it kind of depends how you look at it. If you look at what's in the food, the precursors of the ketones are there, fats, but the ketones themselves aren't. It's interesting. Ketogenic diets will increase ketones because the ketogenic diets is high in fat and low in carbs, and so the glucose in your liver is relatively low, and there's plenty of fats to produce the ketones. But you can also ingest beta-hydroxybutyrate, the ketone, BHB. And a colleague of mine who's deceased now, Richard Veach, he was at the NIH, and he developed what's called a ketone ester, which is BHB, the ketone, coupled to a small molecule called butane. And you can eat this if you're a mouse, rat, or human, and it'll elevate your ketone levels up into what's seen actually with extended fasting, up into like millimolar range, which is pretty high. And then he collaborated with my lab. We showed this ketone ester was beneficial in our mouse model of Alzheimer's disease. And then he collaborated with Kieran Clark at Oxford University in the UK, who's an exercise physiologist. And they showed, first in rats and then in elite British cyclists, that this ketone ester would improve endurance performance. And then now, the ketone is marketed by several different companies, and one of their main targets is athletes. But anyway, there are studies going on. Steve Cunnane, which I've had, by the way, I have a podcast. It's called Brain Ponderings. It's neuroscience.
Right. And hopefully I'll be on that soon. So yes.
Yeah. But anyway, Steve Cunnane up in Canada, in Vancouver, he does PET imaging, positron emission tomography, where you have a radioactive molecule, you inject it in the vein of a person, it circulates, and you can see where that radioactive molecule goes and how much accumulates there. So, for example,
to measure brain activity, sure.
To measure glucose utilization, you have radio-labeled glucose, or different form called 2-deoxyglucose. But he didn't radio-label BHB, but he radio-labeled acetoacetate, the other ketone. And he found that in people with mild cognitive impairment and early Alzheimer's disease, there's a marked reduction in the ability of the nerve cells in the brain to take up glucose, but they can still take up the ketone. And there's multiple clinical trials ongoing. He actually did one clinical trial with what's called medium chain triglycerides, which are the fatty acid precursors to ketones. So you can buy this MCT oil, which is this coconut oil, and it will elevate your ketones. He found that was beneficial, at least within a period of a few months in patients with cognitive impairment. Yeah, I guess the point there is that emerging evidence suggests that elevation of ketones alone can have beneficial effects. I doubt over the long term they're as much as exercise or intermittent fasting, because you may not have this metabolic switching, but you may still get some of the benefits that are due to the ketone component of fasting or extended exercise.
Can I ask you along those lines, kind of the timing, if you are going to try supplementing or taking in exogenous ketone bodies, whether it's BHB or something else, what is the timing? Should you be doing that? Does it matter the time of the day? Does it matter if you take it at the same time you're eating a meal that's high in glucose? Should you only do it when you're fasting or exercising? Or, yeah, I mean, and obviously we don't have all the answers, but I would love your suspicion at this point.
Yeah, it would be a speculation and suspicion because the data aren't there. Those particular studies haven't been done. My suspicion is that, so with these short-term fasts, like 18 hours a day like I do, or something called 5-2 intermittent fasting, which was actually developed by, we helped in a study done in the UK by Michelle Harvey, who found that in 100 overweight women, she randomly assigned them to either 2 days a week eating only 600 calories a day, so that's 5-2 intermittent fasting, or the control was not just eat as they normally would. It was the women in the control group ate breakfast, lunch, and dinner, but they had about 20 to 25% fewer calories than they normally would eat. And over periods of weeks and months, both groups consumed the same amount of calories, lost the same amount of weight. And this was a 6-month study, but the women on 5-2 intermittent fasting had a greater improvement in glucose, insulin sensitivity. And anyway, this is actually, I keep getting off track, but historically is very interesting. Then what happened, we published that paper in a good journal with Dr. Harvey, and a producer at the BBC, Michael Mosley, picked up on it. And he did a documentary for the BBC called Eat Fast, Live Longer. And that aired in 2013. And then that kind of went viral first in the UK and then kind of spread on the internet. And that was really what led to the kind of explosion of chatter on the internet about intermittent fasting, because before then, it was mostly just, the science that was being done on it and not much in the popular press. So
what I was asking is, though, does it make sense if you are going to try these exogenous ketones like BHB, should you do a pre-workout? Should you do it mid-morning? Should you do it after a meal? We don't have the data in humans, but
my guess is it's better to do it during the fasting period, because during these short fasts, like I mentioned,
right,
18 hours a day or on those 2 days, 5-2, the ketones only go up into several hundred micromolar. So that's 0.2 of a millimolar, right? Whereas if you do long-term fast, or if you take the ketone ester, they'll go up to a couple millimolar, right? So if you take, the other thing is if you take the ketone ester and you're eating carbs, the carbs may preclude the switch of neurons in the brain from using glucose to ketones, because they may then be preferential to using the glucose that's there before they'll use the ketones.
We talked about switching from the brain using glucose to using ketones and the switch. Do you have any sense on how long you should go? I mean, obviously there are people out there who can, I've done a water fast for 10 days. There are people under clinical studies can do it longer, but ideally, how often do you want to switch from using ketones to glucose? Again, the cool thing about our metabolism is it's flexible, but what do you think the ideal flexibility would be?
Dr. Josh, we don't know. It's interesting. So my lab focused mainly on the every other day fasting, and then the 5-2 and the clinical studies, and then other labs have focused on daily time-restricted eating, like I do. For example, Sachin Pandit, the Salk Institute. And so the clinical trials, and then Walter Longo, he came up with this other idea, 5 days a month, eat only like whatever,
right,
500
right,
or 600
right.
calories.
I just interviewed him. That episode should come out shortly. Yes.
Yeah.
He's amazing.
And so kind of each of the prominent groups in the area, you started with something and they stick with it. And there's been no direct comparison in the same study, the same population of people looking at the same endpoint. So ideally, you'd want to have daily controlled and daily time restricted eating and 5-2 or every other day or 5 days a month, all in the same study. But we do know that compared to the control, which is breakfast, lunch, dinner, and all of these are better. And as far as the timing, now the long-term fasts, the groups that really done a lot there is in Germany, actually, at the Buschinger clinics, where people go. There's like a resort, mainly for wealthy people, actually, that can afford it. But they go there for a couple weeks and they fast for 10 days to 2 weeks in a controlled environment with a lot of group activities and so on. And they have a lot of data, measuring various things, a lot of blood work or just whatever, body fat, so on, before they start on the fast. So they come to the clinic, they do all these measurements. And then at the end of the 10 days or 2 weeks, what isn't available or in very limited amounts is, if these people, they do this only once a year usually, so then they go back home and they go back to their normal eating pattern. My prediction is, 10 days or 2 weeks out of the year is going to be much less benefit than daily time restricted eating or, 5-2 throughout the year or the 5 days a month throughout the year, because you're only having the metabolic switching so I think the back and forth, kind of like exercise, same with exercise. If you exercise for 10 days a year, right, it's not going to benefit you much. It's better benefit but you can the reason that exercise and intermittent fasting, the short-term fast, are so safe is because they're so evolutionarily common in the history of evolution of animals, including our human ancestors, right? And so our system has evolved not only to be able to cope with, not eating for a couple days or half a day or 3/4 of a day, or, having to spend a lot of energy, a few days a week to track down, kill. But our systems have evolved to function very well with that eating pattern and exercise pattern, if you will. And so other patterns that aren't evolutionarily common are not so good.
Right. And if you just look at modern society, most people I meet have never tried fasting. I mean, you see people on an airplane, if they have to go a couple hours without eating, they go crazy.
Yeah, the thing here, Dr. Joss, is that we found, and others finding, that it takes several weeks to a month to adapt to the new eating pattern, intermittent fasting eating pattern. So if you normally eat breakfast, lunch, and dinner, and tomorrow, and you say, okay, tomorrow I'm going to start daily time restricted eating, I'm going to skip breakfast. You're going to feel very hungry and irritable in the morning the next day because you didn't eat breakfast, because your neuroendocrine systems that control hunger and mood and so on, they're adapted to that eating pattern. And we found in animals, and this seems to be true from the clinical studies in humans, it takes several weeks to a month. Again, I go back to the exercise analogy.
Sure.
If you've been sedentary for years and you go out and run 5 miles, well, you probably can't run 5 miles. You'd have to stop. And some people never get into an exercise routine because they feel discouraged in the beginning, right? I'm not feeling good because, it's a big shock to my system, if you will. But you can build up, just as you can build up to exercise over a period of weeks and a month or a couple months and get in shape. It's possible to build up the intermittent fasting, I guess. You could, like, if the idea is you eventually want to skip breakfast and work out at midday like I do before you eat, then you could just kind of advance, start eating breakfast a little later or something like that over a period of a couple months. On the other hand, it's very safe to go cold turkey, not eating breakfast. It's not going to hurt you. And if you can stick with it for a few weeks, you'll no longer be hungry in the morning. And I should also point out that people with obesity often have something called leptin resistance. When you eat a meal, there's a hormone released into the blood, actually from your fat cells. And it goes to your hypothalamus in your brain and tells you, okay, you've eaten enough, you're feeling full, you don't have to eat anymore. So leptin resistance is when your brain does not respond to the leptin, and so you continue to perceive you're hungry when other people with leptin resistance, for example, someone healthy weight, doesn't. So that's why people with obesity often perceive they're hungry when, clearly they have a lot of energy reserves. They're not starving, they're hungry.
That makes perfect sense. Going back to the BHB ketone discussion, one question I had is, it's different if your body's producing ketones from your fat as a fuel, as opposed to if you're taking it in from a drink. And one of the differences is the stimulation of insulin. So what are your thoughts about that?
Yeah, well, for example, I mentioned during I didn't mention it. During fasting, certain neural circuits in your brain become more active, particularly those involved in attention and learning and memory, which kind of makes sense. You're trying to figure out where to get food, again, from the evolutionary perspective. So that increase in neural activity seems not to involve the ketone, BHB. There's something else going on there. But nevertheless, that increase in activity, we think, is important for some aspects of the beneficial effects of fasting on brain health. And there's good evidence that people who keep their brain intellectually challenged throughout their adult life have better brain health outcomes, are less likely to develop dementia and Alzheimer's disease. And so that's the use it or lose it idea. And we know a lot of things that are going on with more activity in your brain. One is an increase in BDNF, but there are many other things going on that, for example, something I didn't mention, I mentioned in relation to muscle. So the number of mitochondrial cells can be increased. Okay, and in muscle exercise, intermittent exercise will increase the number of mitochondria in each muscle cell. So there's more energy, more ATP the muscle can produce to keep the muscles functioning longer or stronger. And same is true in your brain. When your neurons are more active over time with repeated challenge recovery cycles, so intellectual challenges during the day, good sleep at night. Sleep's probably the most important recovery period, certainly for your brain. Then the number of mitochondria will increase in your neurons, and even number of synapses between them will increase.
Wow. All right. So you've been on the cutting edge of Alzheimer's research, fasting research for a very long time. What's exciting to you in the new technologies or new results that you have maybe heard of or seen that you think will be coming up in the next 6 months, for example?
Yeah. Technologies or dietary supplements or drugs, not much. Maybe ketone ester.
Okay, that sounds good.
As you know, in our country that we live in, and that as of not a few months ago, as of January, we're investing less in is our healthcare system. So the way our system is organized, and you know this probably as well as anybody who's a practicing MD, is let people get sick, and in what you're doing now, diet and lifestyle and things you can, risk reduction, prevention, kind of optimizing health throughout life. Our system is geared, it's for profit, it's geared for letting people get sick and then treating their symptoms. Pharmaceutical companies, that's what they're doing. They're focused on treating disease, and the diseases they're treating are mainly things caused by poor diet and lifestyle, or at least facilitated by that. Cardiovascular disease, cancers, stroke, we think Alzheimer's disease, and actually any chronic inflammatory disorder, arthritis, So I think the emphasis has to be on education and somehow getting for-profit drivers of poor health. So I mentioned pharmaceutical industry, but also agricultural industry, unfortunately, is geared towards monoculture. So you have corn, soybeans. The corn you use to feed meat animals, like kind of red meat that, a lot of consumption isn't really good for you. It's pretty good evidence for that. And they use the corn to make high fructose corn syrup.
Right, which is toxic to your liver and brain, yeah.
It turns out it's possible to make ketone, beta-hydroxybutyrate, from corn
Wow.
through a fermentation process. Richard Veitch, who was this colleague at NIH, he passed away. He was well into his mid or late 80s. recently, but he tried to get, I can't remember, he tried to get two big ag industries to buy into this idea of, essentially switching from producing high fructose corn syrup to producing this ketone from the corn.
Right,
that
which would
still
be,
may happen.
which actually would be beneficial as opposed to toxic.
Yeah. So I think that the big efforts have to be up front, and people have to take self-initiative to do what they can do now that we know work. And for the brain, it's just three things: regular physical exercise, moderation in calorie intake, and intermittent fasting, and keeping your brain intellectually and socially engaged. And reducing bad stress or chronic psychosocial stress, getting plenty of sleep, and having good diet composition. My view is plant-based diets are the best. Avoid simple sugars,
Right.
but plant-based. I think whole grains are okay.
Right.
And then a lot of fruits, vegetables. It turns out, I wrote an article for Scientific American in 2015, came out. It was called What Doesn't Kill You, but the idea was that the chemicals in fruits and vegetables that are good for our health, the reason those chemicals are in the fruits and vegetables from the plant's perspective, evolutionary perspective, is to keep insects and animals from eating too much of the vital parts of the plants. So these are kind of natural noxious agents. So everything that people will find on the internet, and then there's evidence for some of these, like sulforaphane in broccoli, or curcumin in turmeric root, or I don't know, there's a whole bunch of them, even caffeine, actually. If you take the pure chemical and put it on your tongue, it's very bitter tasting. So these are concentrated in the skin of a fruit, or the vital parts of we discovered this one and worked with this one that's in tomatoes. It's called tomatidine. It's a chemical that's very high in the skin of the green tomatoes. And as they ripen and turn red, its concentration in the skin greatly is reduced. So the insects won't eat the green tomatoes, right? But they'll eat the red ones. And so this chemical, we think, is keeping the insect from eating the green tomato. But then we did studies in mice and cultured neurons that had some beneficial effects if you have the right dose. Anyway, there's a whole another story on these phytochemicals and how they may be acting and how our body deals with them. It's a very interesting story.
That's beautiful. And I wish we had hours more, and maybe we'll do this again. But Dr. Mattson,
maybe we can do one on the dietary phytochemicals. I'll send you a couple articles.
I would love that, and I'll share that in the notes. And let me just find out though, if people, the audience, they want to reach out to you, contact you, are you on social media? How can they find you?
Email is the best. I'm old school. I never got on social media when it first came out. I just kind of watched for maybe a year, and I said, this is going to be a waste of time. A lot of gossip on there. Now I'm too busy to spend my time on that. I've got better things to do. So my email is at Johns Hopkins University, where I have since I retired from the NIH, I'm an adjunct professor at Hopkins. I teach a couple courses for their neuroscience program. So you should be, if you just put Mark Mattson, Johns Hopkins, the page will come up. It'll have my email address. It also has my phone. Don't call me. Send email if there's something that's worth communicating further. I can talk on Zoom or something. But
that's all. And it's Mattson with 2 T's. Yeah. Awesome. So, Dr. Mattson, thank you so much, not just for your decades of research in this field, but also for being willing to share that with the audience and with us today. I really appreciate everything you've done for advancing human science. And this is real, because if it weren't for you, there's very little clinical research on fasting in humans. And you're part of a very limited but very important area of research in neurobiology involving Alzheimer's. So I salute you, and I really appreciate your taking the time today with us.
Well, I really appreciate your compliments, and I've enjoyed my career as a scientist. It's a lot of fun, a lot of hard work, and I used to coach high school cross country, and my kind of mantra to the kids on the cross country team was, run hard and have fun. And so that's kind of what I applied to science. It can be both hard work and fun. And same with health. It can be both hard work and fun, right? And it can both be challenging and then relaxing during the recovery period.
Well, yeah.
And and thank you for what you do, Dr. Josh, getting, authoritative information out there, talking with people who are doing respected work. And then, I think, again, people need to be able to critically evaluate things. And I think to do that, you have to actually go to the science and not get everything, third hand, fourth hand, particularly by people who aren't experts in the field.
Exactly. You have to talk to the researchers like you, and you actually have to read the original papers, not rely on third parties.
Yeah.
Awesome.
Okay.
Peace and plants. All right.